Good afternoon, everyone, and thank you for joining us today. My name is Chris Grant, and I'm pleased to welcome you to Annovis Bio Incorporated's Investor Webcast. Today, we'll be discussing the detailed findings from the recent Phase 2/3 study of Annovis Bio's lead drug candidate, Buntanetap. Before we begin, I'd like to remind everyone that today's call may include forward-looking statements. These statements are based on current expectations and assumptions and are subject to risks and uncertainties that could cause actual results to differ materially from those projected. For more information on the risks and uncertainties related to Annovis Bio's business, please refer to the forward-looking statements in the company's latest SEC filings. To guide us through today's presentation, I am pleased to introduce our speaker, Dr. Maria, the founder, president, and CEO of Annovis Bio Incorporated. Dr. Maria has been a pioneer in the field of neurodegenerative disease research, and her leadership has been instrumental in driving the company's mission forward. I would also like to add that during the webcast, participants will see the Q&A icon in their taskbar. By clicking on this icon, you can type in your questions and submit them with your name attached. We'll be addressing those later. And without further ado, I will turn things over to Dr. Maria.
Hi, thank you very much, Chris. Good afternoon, and thanks for joining us. So I've been calling this meeting for two reasons. One is there are a lot of questions as to Alzheimer data that we released a little while ago. And two, we have some additional data on ApoE4. So what I'd like to do today is explain what the study was that we conducted. It really was a dose-ranging study. What we found, how we were stuck with patients that didn't have Alzheimer's disease, how we actually eliminated them, why we had to resort to a post-hoc analysis, and how we saw highly statistical data in cognition in early Alzheimer patients. The next part of the study then goes to ApoE4. Why are we interested in ApoE4? I will get into that a little bit later. But basically, ApoE4 is not just a risk factor for Alzheimer's disease. It is a genetic predisposition. That was just published a few weeks ago. And also, ApoE4 patients have an issue with the existing drugs on the market. So let me go into the study, and the cursor doesn't move, okay? Next. The forward-looking statements, Chris already read them. This is what I'm going to tell you today. And this is the study. So we conducted a randomized, double-blind, placebo-controlled dose-ranging Phase 2a study. I want to point out that we only had 80 patients per group, and 4 groups, placebo, 7.5, 15, and 30 mg. The reason we did that, we knew that with 80 patients per group, we were reasonably to very sure we would get an ADAS-Cog readout. We really weren't sure we were going to get a CGIC or an ADL readout, but we figured that if we see a trend, we can then use it in our next study. So the inclusion criteria, and this is important, we used the National Institute of Aging criteria for aging and Alzheimer's disease. These criteria were put in place in 2011. As you know, or most of you know, pharma today, Leqembi, Aduhelm, donanemab, they have used PET scans and MRIs to select patients to go into their study. That assures that these patients have plaques and tangles. It assures they have Alzheimer's disease. We figured that was expensive, and this was a smallish study, so we didn't do that. In hindsight, that was a mistake. But, you have to live with your mistakes. So we took age 55 to 85 and MMSE 14 to 24. Again, we increased the MMSE from our Phase 2a study where we knew it worked between 20 and 28. So we figured we're going to try in 14 to 20. The key clinical outcomes, we really structured them according to the FDA. The FDA wants you to have two co-primary endpoints. One is cognition, that's the ADS-COG. The other is a functional study. Be that the clinical global impression of change or the activities of daily living. Both studies need a lot of patients to show statistical significance. I hope we were going to get a trend. So with that study, this is the data we got 6 months later. If you look at difference from baseline, every group in this study is highly statistically better than baseline. Unfortunately, placebo is highly statistically better than baseline. When we then deducted the treated patients from the placebo patients, we had no statistical significance. And that is when we resorted to what we had learned. In November, T3D, which is a private company, so you probably didn't hear about them, said that 60% of their patients did not have Alzheimer's disease. And in December, BioWe said that 80% of their patients didn't have Alzheimer's disease. This was very scary. When we looked at our ITT population, we saw that some patients went from having an MMSE of 14 to having an MMSE of 28. So they went from being severely disabled to normal. That's a miracle. And we suspected they didn't have Alzheimer's disease, but we obviously didn't know. Now we sent our blood samples to C2N, which in the meantime had come up with two plasma level tests. One is a p-tau-tau ratio. The other is an A-beta 40-42 ratio. Both ratios represent plaques, mostly the A-beta, and the tau ratio represents plaques and some tangles. C2N has published that the tau ratio is more sensitive for Alzheimer's disease than the A-beta ratio, so we use that ratio. As you can see, we started subdividing our people into Alzheimer patients because they had the correct p-tau to tau ratio, and we came up with 40% of our patients that did not have Alzheimer's disease. We then further subdivided into moderate Alzheimer patients and early Alzheimer patients, and there we saw that 55% were moderate and 45% were early. When we looked at the ADS-COG of those two populations, on the left, you see the moderate ones. The moderate ones show a minor improvement with the 15 mg group, but the placebo does fantastically better. So again, there is no statistical significance. Now, in hindsight, this really doesn't make sense. If you have a high placebo effect, you should see it all over. Why only placebo does so much better, we don't know. We're going to go back and look at that data more closely. However, when we looked at the MMSE 21 to 24, which is the early Alzheimer patients, we saw beautiful, statistically and highly statistically significant dose response that is significant from baseline as well as from placebo. So we knew our drug works in early Alzheimer patients. Well, the next question then was, okay, it works in early Alzheimer patients. That's not so different from other drugs, even though I'm going to show you later how it differentiates itself. But we thought, how does it differentiate itself not in terms of efficacy, but in terms of APOE4 carriers? APOE4 carriers, as I said, have a much higher prevalence of Alzheimer's disease. APOE4 get Alzheimer's 100% of the time. APOE4 3 or 2 get it a lot, maybe not 100, but 70 to 80%. So also, APOE4 carriers don't respond very well to antibodies against A-beta. Neither Aduhelm nor Leqembi nor donanemab work very well in APOE4. They show a very small improvement. Improvement means less decrease in cognition. However, on top of that, APOE4 carriers are much more susceptible to ARIA. In fact, all the patients that died in the 3 antibody studies were APOE4 carriers. So they have less efficacy and more safety issue. ARIA is brain bleeding and brain swelling. Well, in fact, the FDA told all 3, well, I'm not sure yet about donanemab, but they probably will also have a black box saying that APOE4 patients need to be very careful if and when put on any of these 3 antibodies. So it was important to us to see whether our drug is also not very efficacious, but very toxic in APOE4, and that's what we checked. So on the left, you see the data I just showed you in early Alzheimer patients. Again, you see that at the best dose, our drug improves cognition by 3.3 points. If you look on the right, we subdivided into APOE4 carriers, and our drug improves by 3.15 points. That's pretty much the same. The reason we don't have statistical significance from placebo in the APOE4 carriers, we have a lot less patients. We have lost a third of our patients by subdividing into APOE4 carriers and non-carriers. But the data looks pretty much the same. Buntanetap is as efficacious in APOE4 carriers as non-carriers. So how does it look with safety? Because we had absolutely no deaths. We had no serious drug-related adverse events. So we looked at all treatment-related adverse events and treatment-related study reverse events, and then serious treatment-related events and serious study-related events in APOE and in non-APOE carriers. And what we saw that all across the 3 doses as well as placebo, we see pretty much the exact same safety. Our drug is safe independent of whether in APOE4 carriers or non-carriers. The only 3 serious treatment-related adverse events, you see them in placebo, 3 in the first table. And of course, they're random. When you have these few people, it's easy to have 3 serious events, but they're random and they were not drug-related. APOE4 carriers and non-carriers show the exact same safety profile. They show the same side effect profile. And so we know that our drug does not just work the same in APOE4 and carriers and non-carriers, but is also safe in APOE4 carriers and non-carriers. That makes it a very suitable drug to be used as in co-administration with other drugs because it doesn't increase their toxicity, but it increases their efficacy. Now, how does our drug compare to the drugs that are on the market? In red, you see a symptomatic drug. Aricept or donepezil has been on the market for over 20 years. It's a generic, and it's given to most people that go to a doctor and say, I'm getting forgetful. It's not working terribly well, but it's the best we have. If you look at 3 months, I showed 3 months because that's the length of our study, so we can compare to our study. But if you look at 3 months, they show an improvement of 1.5 points, and at 6 months, an improvement of 2 points. That is about 2 points better than placebo. If you now go to Buntanetap, the 3 lines on top in purple, you see that at 1 month, we see a 4.4 point improvement. At 3 months in early Alzheimer patients, we see a 3.3 point improvement. And in moderate, a 0.65 point improvement. I want to point out that the difference from placebo in the 2 studies that worked, which is early Alzheimer patients, is between 2.5 and 3 points. I just told you that Aricept, the difference between placebo and treated, is 1.5 to 2 points, so less. Our drug is about twice as good, if not 3 times as good as Aricept. The reason I say maybe 3 times as good is we are giving it on top of Aricept. Patients on our drug already have the 1.5 point improvement, and then they take our drug, and then they get another 3 to 4 point improvement. So that's symptomatic. At this point, our drug looks like a really good symptomatic drug. Now, we always want to talk about disease modification, and we'll talk about that. But at this point, we only have a 3-month study, and it looks like a really, really good symptomatic drug. So let's look at the 2 blue lines at the bottom. One is Leqembi, and the other is donanemab. Leqembi was approved last year. Donanemab was not approved, but recommended for approval yesterday. And in fact, if you look at 3 months, I have the Leqembi data. I don't have the donanemab data. Leqembi at 3 months does pretty much nothing. It's very similar to baseline. If I look at 1.5 years, Leqembi worsens cognition by over 4 points. And it's only 1.44 points better than placebo. So the spread between how much placebo worsens and Leqembi worsens is not very high. Eli Lilly's donanemab, as I said, I don't have 3 months, but again, at 1.5 years, it worsens cognition by 3.17 points, and it has a 1.5 point spread from placebo. I want to go back to that spread, because that's the spread we see in Aricept. We have a much bigger spread. Another thing I want to point out in this table is the patient population. Our drug didn't work in 14 to 20. None of these drugs work in 14 to 20. If you look, E-size, Aricept, 21 to 26. E-size, Leqembi, 22 to 30. Eli Lilly's donanemab, 22 to 28. And in fact, as far as I know, there is not one drug that works in more moderate patients. So maybe we were too aggressive to think we could do something in moderate, but it's good to know. So what does it all mean? Sometimes I think we all don't quite understand what this disease modification and this symptomatic has. If Aricept improves cognition and Leqembi doesn't, why is Leqembi better than Aricept? And this shows it. The red line is Aricept. It improves cognition, but after about 6 months, the efficacy fades and the slope goes downhill fast and eventually reaches the regular course of the disease, which is the yellow line. If people take nothing, that is how their cognition goes away. Now, the blue line is a disease-modifying drug. That could be donanemab, or it could be Leqembi, or it could be Aduhelm. They're all similar. What they do, they don't improve cognition, but they slow the progression. The way that is measured is the slope of the blue line is slower or flatter than the slope of the yellow line. There is not one drug I know of that stops the disease. That would be the green line. That would be a great drug. Now, on top, I have a purple line, and I am going to go way out on a limb here, so take all the legal excuses that I don't know what I'm talking about. What you see right now, what we see right now, is that the purple line is better than the red line. It definitely is. We saw it in 2 studies. There is no limp here. I'm sure it is statistically significant. However, what does it do after 3 months? That is the part we don't know. If it is symptomatic, it will drop just like the red line and eventually go down to yellow. If it is disease modifying, then the slope of the purple line, which will be slower than the yellow line, And in fact, the way we drew this is that the purple line has the same slope as the blue line, so that it has a disease-modifying effect. Now, granted, we haven't proven that yet. We really haven't, but that's exactly our next step. We want to show that Buntanetap improves cognition in the beginning and then slows the decay of cognition. If we can do that, then we have proven that there is a drug that is symptomatic and disease-modifying. Right now, most people think that's not possible, that you either have a symptomatic drug or you have a disease-modifying drug. I don't know. I will tell you in a few years. So where are we going from here? We did the study on the left, the mild to moderate 3-month symptomatic study, where we found that we have to be in early Alzheimer patients, not in advanced. And we are just summarizing all the data to submit a request for a meeting to the FDA. The meeting will entail one major request. The request is that we can do a 6-month symptomatic / 18 months disease-modifying study. And the reason we want to do that is if we can put them together, we'll start a large study with something like 600, 700 patients. And after 6 months, we analyze the data. And we can file an NDA for symptomatic because we will have reproduced the data we have in the 3-month study in a 6-month study. And we will know if at 6 months, it is still better than Aricept. It is still as good as at 3 months. However, we continue the study. And after 18 months, then we will file the NDA for disease modification. So in one large study, we want to do symptomatic and disease modifying. If everything goes according to plan, and we get the FDA meeting in September, we'll start the study in winter, and by the end of next year, we will actually have the data of the repeat symptomatic study, and we can start filing an NDA for symptomatic. We'll also have an open label study because all our patients liked our drug and want to go back into the study. So I think we are in a good place. Granted, we didn't make the primary endpoint because we didn't know. We didn't know that some people fake having Alzheimer's. We learned it too late. We didn't know that the drug didn't fit or didn't work in more advanced patients and moderate patients, but we know now. And the data is really solid. The data we have in early Alzheimer patients is really solid, and it actually totally reproduces the data we had in our phase 2A study in early Alzheimer patients. So that's all I wanted to say. We will give you a lot of time for question and answer because I know you have a lot of questions, and I'm here to answer them all. Thank you very much.
Thank you, Dr. Maria, for that insightful presentation and for sharing the exciting findings from your phase 2, phase 3 study. We are now in the Q&A portion of the webcast. Before we begin, I'd like to remind everyone that if you have a question, please use the Q&A feature in this Zoom meeting, Zoom webinar. We will do our best to address as many questions as possible, and we'll try to, some of them are duplicates, so we're going to try to address them all at the same time here. But at the same time, let's go ahead and queue them up. Go ahead and put your questions now. Oh, there they go. Just take a moment. All right. And our first question comes from David. Can you be more specific about your future plans for Alzheimer's disease?
Well, yes and no. Obviously, we want to discuss the study with the FDA, but I can tell you what we plan to send to them. We plan to send to them a kind of simple-minded study, if you want to put it this way. It's in early Alzheimer's patients. We will do the tau ratio before we include them into the study. We will also do volumetric MRI. We will take plasma samples before, and during and at the end, same for volumetric MRI, we will measure A-beta and tau. Because we saw in our phase 2 study where we have already measured it, that we actually affect them and tau goes down. So that's pretty much the inclusion criteria. So we will make sure they're early Alzheimer patients. The MMSE will be 22 to 30 because from our phase 2A, we know that it works all the way up to less impaired patients. And it's an 18-month study with a 6 months analysis.
And our next question comes from William. Can you discuss the continued development of your drug for Parkinson's disease?
Well, the two questions are kind of similar because the drug is the same drug. We do believe it acts the same way. And so, yes, we don't have the Parkinson's data quite yet, but we basically will do the same thing. We'll go to the FDA, we'll summarize the data, and we'll ask them, because the Parkinson's study was a 6-month study, so we'll ask them to let us combine a symptomatic study with a disease-modifying study so that we, again, at 6 months, we will analyze the data for symptomatic, and at 18 months, we will analyze it for disease modification. In Parkinson's, one of the issues is the UPDRS2, and it's hard to get because, as far as I know, no company to date has been able to show an improvement in UPDRS2. So we're really curious to see whether our drug shows something in UPDRS2. But then realistically, maybe in Parkinson's, we're not using enough patients. Because in Alzheimer's, in order to see a statistical significance in CGIC or ADL, Leqembi used 800 patients per group. Lilly used 1,000 patients. Well, we're using 80. That's not enough. So in Alzheimer's, we want to go to 600 because maybe that allows us to see that. In Parkinson's, most studies, because they're usually not disease modifying, in Parkinson's, most studies will do L-DOPA have been symptomatic. So people have not really used large numbers of patients. And once we know what our data is in the phase 3 that we are going to report in a few weeks, we may decide to also do a 600-month study so that we have enough patients to see something in a UPDRS2.
Excellent. Very good. And from Zoe is our next question. The Alzheimer's study failed to meet its primary endpoint. Why do you believe that the FDA will consider it a symptomatic study?
Well, yes, it did fail the primary endpoint, and it's not a pivotal study. What we are telling the FDA, and what I believe the FDA will see, that this is a symptomatic study on steroids. The data for symptomatic is really, really good. Okay, I want to show disease modification. I know that. But just think, even if it is just for symptomatic, you could give it on top of Leqembi. Leqembi isn't improving cognition. It slows the decline. We gave it on top of Aricept. It actually improves what Aricept can do. You can give it on top of anything. And you can give it without any other drug because it has absolutely no side effects. So I hope it has disease modification, and it will show that in the 18-month study. But even just as a symptomatic drug, it is 3 times better than what we have right now. So I do believe the FDA will see that. And when we ask them to be able to reproduce a symptomatic study included into an 18-month study, I believe they're going to say yes. And that will really allow us to have the data of the symptomatic study by the end of next year.
Excellent. And thank you all for your questions and for participating in today's webcast. If we didn't get to your question, or if you have further inquiries, please feel free to contact our investor relations team at ir@annovisbio.com. Thank you once again for joining us today, We appreciate your continued support and interest in Annovis. Have a great day, everyone.